Hi, welcome back to Chapter 15. This chapter's title is The Future Isn't What It Used to Be, Biotechnology. First up, let's look at what is biotechnology. So when we use the term biotechnology, we are talking about the use of technology to control biological processes as a means of meeting societal needs. So we are using technology and essentially manipulating living things. So let's look before we begin this chapter at the difference of what can we do in biology and biotechnology and what should we do. So many of the concepts in this chapter are very controversial. We'll look at things like using embryos to get newly fertilized embryos to get stem cells that offer various treatments. And we'll look at using biotechnology to modify foods, to change the foods to have characteristics that we like. And all of these, as well as using DNA evidence from crime scenes to convict or release certain people. So all of these ideas are a good illustration of the importance of the idea in the field of science that we need to look at not only what can we do, but what should we do. So science, there's a lot of things that we can do, and but we need to not just look at in science what can we do, but what is ethically right and what is healthy and what should be done. So these topics are often very controversial in terms of ethics. So what is the right thing to do in terms of health safety? So if we're modifying foods or making drugs, is this safe for our body? And a big issue that comes up is, do the ends justify the means? So is the method that we're using, is the outcome of that justified by that method? And that's going to be different. Different people will view that very differently based on their background and beliefs. So there's a lot of controversial things, but on the other hand, biotechnology can and has been used to study DNA at crime scenes and release innocent prisoners, also used to convict guilty people. So there are a lot of good things that can come out of biotechnology, but just give you a heads up as we start the chapter, there are a lot of things that are very controversial as well. So I just want to point out that this is I'll go through here the lecture for chapter 15, but there are also a lot of other videos to watch related to topics that we discuss in the lecture, and the links are located on the chapter 15 lecture page. So when you're done with this lecture at some point this week or next week, go back and watch those video links. They are all pretty short video links. So there'll be a couple questions on your final from those videos as well as from this lecture. So section 15.2 is on transgenic biotechnology. The term transgenic means when we talk about a transgenic organism, it's an organism whose genome has stably incorporated one or more genes from another species. So it's mixing the genes from one species, so a segment of DNA, into another species. And many biotechnology products are produced within transgenic organisms. So we'll look at an example of this on the next slide. So you may be thinking at this point, why on earth would we want to take the DNA from one species and add it into another species? So let's look at an example where that's done. So this is done to produce something called the human growth hormone, and let's go over what that is and why that's important first. So the human growth hormone is a hormone produced by the pituitary gland. And in children, if you have reduced levels of human growth hormone, which can be due to various causes, one could be a genetic cause, but also due to other causes. Children who have reduced levels of human growth hormone will look much younger than other children their age. They will have a chubby body build, impaired hair growth, delayed puberty, or short stature. And these conditions can be improved if they take human growth hormone to lead to a more standard level of growth throughout their life and reduce these other symptoms as well. Adults can have a lack of this hormone due to damage to the pituitary gland. This can happen after radiation therapy, head injury, or certain infections like meningitis. And in adults that have damage to the pituitary gland and lower levels of human growth hormone, their symptoms can be anxiety and depression, increased fat around the waist, increased risk of heart disease and stroke, weak heart, weak muscles and bones, tiredness, reduced ability to think, and other conditions. So some pretty serious side effects can come from having a lack of this hormone. You can have serious side effects from having a lack of any hormone, but looking at this example, there's definitely benefits to being able to get this hormone into your body when you have these conditions. The way this is done is by taking human growth hormone from a human and incorporating it into a bacterial cell. We'll go over how that's done. But you take this human growth hormone gene, you put it into a bacterial cell, and then that bacterial cell will then produce human growth hormone, which can be collected and given to humans as a form of a hormonal replacement therapy. So the way this is done is a human pituitary gland cell has a gene to code for the human growth hormone. And that specific segment of DNA, that gene is removed, so it's cut, the ends of the DNA are cut at specific points. That DNA segment that codes for producing human growth hormone is then, after it's removed, it's placed into a loop of bacterial DNA. So bacteria can have a loop of DNA in them called a plasmid. If you think of your bacteria, it's a long straight line. They can have bacteria in this loop called a plasmid. So this plasmid is cut. So it's cut. So we take this bacterial loop open, or we take this bacterial loop of DNA, cut it, and then take the human growth hormone DNA and put it into that loop of bacterial DNA. You now have a new loop that contains human DNA and the rest of the bacterial DNA. You can put that loop, then insert it back into the bacterial cell, and then the bacterial cell will then produce human growth hormone. And every new cell that divides from that will now divide and replicate this DNA. So it won't replicate their old DNA or replicate the new DNA with the bacterial growth hormone. So you don't have to do this process of splicing the gene out and adding it in over and over. You do it once. You now have a bacterial cell that will divide and replicate on its own and produce human growth hormone. And then that human growth hormone can then be given to humans. So we now know the overall process of how this happens. The first step is to remove human DNA and then place it into bacterial DNA. Let's look at a little bit more detail on how this is done. So the first thing that we need to do is remove this section of human DNA from the pituitary gland cell. And they use something called a restriction enzyme, that's this term here, which is a protein derived from bacteria that can cut DNA in specific places. So this restriction enzyme recognizes a certain segment of DNA and makes a cut there. So in the case of the human growth hormone, it recognizes the sequence of DNA that it needs to recognize in order to, this is your human growth hormone section of DNA, and on either end of this section of DNA is the code GGA-TCC, and it's on both ends, GGA-TCC. Now, if you'll notice, the corresponding code on the end is also in reverse order, GGA-TCC. So the same code, just in the reverse order. And so the restriction enzyme recognizes this segment and it makes a cut between the two G's of this segment here. It also makes a cut between the two G's of this segment here. So you get a cut here and you get a cut here on the top strand. On the bottom strand, it recognizes the same sequence and makes a cut between the two G's. So it makes a cut here and a cut here. What you're left with now is a loose piece of human DNA that codes for the human growth hormone. Now, the human growth hormone, as we just learned, gets cut at the sequence GGA, TCC. So we have these pieces here that have been cut out of the DNA. The bacterial plasmid also has that same segment of DNA and will get cut at that same place. So it'll get cut in the GGA, TCC segment. It will get cut between the two G's. So it gets cut in the same place. And what you're left with is sort of like two pieces of a puzzle that this G will bind with this C. Remember, G and C bond together. This A will bond with this T. A and T bond together. This T will bond with this A, and this C will bond with this G. So it's like two pieces of puzzle that they're called sticky ends because they want to bond together like two ends of a magnet, two pieces of puzzle. They match together and they will hydrogen bond together And so this piece here that's been removed from the human cell wants to naturally bond to this, not naturally, but due to the bases pairing up, it wants to bond to the bacterial cell here. So when you put the human DNA next to the bacterial DNA and they've both been cut at the same place, they will bond together. And this produces what we call recombinant DNA. Again, a combination of DNA from two different species. Once more, when we say recombinant DNA, we mean two or more segments of DNA that have been combined by humans into a sequence that does not exist in nature. So again, just as a reminder, we take our human DNA, place it into DNA that's been taken out of bacteria that's been cut open. We merge the two DNA together, place that DNA plasmid, this recombinant DNA, back into the bacterial cell, and now every time the bacteria replicates, it produces new copies of the same combined DNA, and all of these bacterial cells will now produce that human growth hormone that can be used to treat humans. So human growth hormone is an example of one type of medicine that's used by this process of transgenic biology. There are a large number of medicines and vaccines today that are produced through transgenic biotechnology. Transgenic organisms that are used for this purpose include not only bacteria, but also yeast, hamster cells, and mammals such as goats. Transgenic food crops are planted in abundance today, also in the United States. Next up in section 15.3, we will talk about reproductive cloning. A clone is a genetically identical copy of a biological entity. Genes can be cloned, as can cells and plants. Reproductive cloning is the process of making adult clones of mammals of a defined genotype. Dolly the sheep, who you can see here with her surrogate mother, was a reproductive clone. Today, reproductive cloning of mammals is carried out through variants of the process that was used with Dolly. We'll go over that process next. The process is called somatic cell nuclear transfer, or SCNT. Let's look at this process of somatic cell nuclear transfer, or SCNT. So what happens here is we have the white sheep that we're going to make a clone of. This is what they did to make Dolly. So they took the white sheep, they took DNA from her udder cell, so one of the udders that produces milk, took DNA out of it, and then they took a cell from the surrogate mother and took the egg cell, removed the nucleus out of it, so took the nucleus out, which is where all the DNA is. They added the DNA from the udder cell of this white sheep into this now empty egg cell, added some electric current. This caused the two cells to fuse together and prompted an activation that reprogrammed the donor cell DNA. This caused the fused cell to start developing as an embryo. After some incubation, the embryo was implanted into a third sheep, which served as the surrogate mother. So this sheep had the surrogate egg cell, and this sheep is where we got the DNA from and made the clone from, and then a third sheep was used to implant the embryo, this now growing embryo, to implant it into this sheep to be the surrogate mother, and Dolly was born. Dolly grew into an adult. Now, Dolly had a lot of health problems, and being the first mammal cloned, this definitely put a negative perception on the idea of cloning. So when Dolly was 1 year old, analysis of her DNA showed that her telomeres were shorter than would be expected for a normal sheep of the same age. Telomeres are these end caps on the DNA molecule that protect the DNA from damage. As you get older, these telomeres naturally get shorter, but her telomeres were a little shorter than would be expected for another sheep of her same age. Dolly got arthritis and then also got lung tumors and was euthanized at age 6. A normal sheep life expectancy of that breed is about 11 to 12 years. So because she died early and had some different health problems, that definitely put a negative spin on could cloning produce healthy organisms from the cloning process. So what they have said currently is that Dolly's lung tumors were the result of a contagious disease that spread through the flock that could have happened to any sheep. And currently, they say that her offspring and clones produced from her all appear to be living normal, healthy lives. So the clones produced from her are about, I think, 9 years old and seem to be still alive and still healthy at this point. The idea has, of course, come up that if we can clone other mammals, we can probably clone humans. If you were to clone a human clone would be a genetic replica of the person who provided the donor DNA cell. So whoever provided the cell and the DNA was extracted from that cell, a clone would be made from that person. The donor and his clone, in terms of genetics, would be genetically identical in the same way that identical twins are identical. They would obviously be different ages, not born at the same time, and but their DNA would be exactly the same. The difference being that identical twins, of course, are through a natural process, and cloning is through an artificial process. As you can imagine, the idea of cloning humans is definitely a very controversial topic. Next up, section 15.4, let's look at cell programming. Most cells in the adult human body have undergone commitment, a developmental process that results in cells whose roles are completely determined. So the cells go through a process where they become, as we say, committed. They only serve a certain function. So your skin cells are going to function as skin cells. Your heart cells will function as heart cells, and those cells perform a specific role, produce specific proteins based on what where that cell is located and what job that cell needs to do. Most muscle cells have undergone commitment, for example, and hence can be nothing but muscle cells and give rise to nothing but muscle. Now, there's one type of cell whose role is not committed yet, and this is the embryonic stem cell. And embryonic stem cells can still become any different type of cell. Now, cells, when an egg is fertilized, it starts to divide. So the one fertilized egg splits and becomes 2 cells and then 4 cells and starts dividing fairly rapidly. Now on day 5 after fertilization of the human embryo, there's this inner cell mass here inside what's called the blastocyst, which is just this group of cells that have begun dividing. So there's this group of cells inside here called the inner cell mass. And cells from the blastocyst inner cell mass are known as embryonic stem cells. So if you hear the term embryonic stem cells, these are cells located within a very young embryo, and they can still give rise to all the different types of cells in the human body. So in the normal process of the cell growing and developing, these cells would continue to grow and divide. Some of these cells, depending on where they're located within the cell mass, some of these would become brain cells, some would become heart cells, some would become skin cells. But at this point here, they are still capable of becoming any different type of cell. People who might benefit from stem cell therapy include people with spinal cord injuries, type 1 diabetes, Parkinson's disease, amyotrophic lateral sclerosis, so ALS, Alzheimer's disease, heart disease, stroke, burns, cancer, and osteoarthritis. So stem cells may have the potential to be grown to become new tissue for use in transplant and regenerative medicine, so helpful in getting an organ transplant. A lot of times we look at nerve damage, spinal cord injuries as being permanent. Embryonic stem cells might have the potential to regrow new nerve cells. So there are a lot of uses that these embryonic stem cells are being researched for in order to help with a lot of diseases and injuries that would normally be life-threatening or certainly debilitating for life. Okay, so let's look at where they get the embryos for embryonic stem cell treatments. So most of these embryos, where they're getting them from is from in vitro fertilization. And the way that this process works is they take the leftover embryonic stem cells from in vitro fertilization and use those to get the stem cells. Let's look at how they do that. So first, they will take many eggs from a female because not all of those eggs will fertilize and work out properly. So they'll get as many eggs as they can from the female, and they will fertilize them by taking a single sperm and inserting it through a tiny little needle into the egg. So they'll place the sperm directly into the egg, and that will the goal being that that sperm would fertilize the egg, and they'll end up with a lot of fertilized eggs. They wait until the eggs are on the fertilized eggs are on day 3 of development. So these are eggs that have been fertilized, and you can see them growing and developing there and moving. So that's the eggs, what they look like on day 3. So they begin to grow. On the 3rd day, the most viable embryos are chosen to be inserted into the female. So they'll look at all of those embryos under a microscope, and they will choose whether to discard them or to keep them. And so they will usually insert a couple of those embryos into the female for in vitro fertilization. But as you can see, there will often be a lot left over that are still living and viable, and so the leftover embryos can be saved for future use, destroyed, or given to science for research or use as embryonic stem cells. So these will keep developing, and they will develop to the point where they can get the stem cells from these new embryos. What constitutes life is a highly debated topic, and so there are people that would say that it's not life until a certain point, and there are people who would say that this the moment that you create sperm and egg together, that this is that it is a living thing and that you should not destroy a living thing. So it's a super controversial topic of using getting stem cells from these embryos. So I think when we look at the big questions here that come up in this debate are there's of course many people that would say that stem cell treatments offer enormous potential. And do they offer enormous potential? I think everyone could agree yes. There are some really devastating diseases and things that can be helped by these stem cells and by the potential of these stem cells. Do they offer the ability to drastically improve people's lives for the better? I think, again, yes, these are some very devastating diseases, but I think the big question for a lot of people comes down to, is it okay to use an embryo to do this? And that's not a question that we can answer in this class. That's definitely a personal question that you have to think about. And so when you see bills voted on and things on using stem cells, this is where that debate comes from, of whether you're taking a life to do this. So since this is so controversial, there are of course people looking at other options of where they can get stem cells from that aren't from destroying a new embryo. Now, adult stem cells are different from embryonic stem cells, and adult stem cells are found in obviously adults and not embryos. They're found in small numbers and various types of tissues in the adult body. These adult cells have demonstrated some ability to differentiate into various types of specialized cells, and hence are the subject of continued research interest. However, adult stem cells do not have the differentiation potential of embryonic stem cells, and they don't have the embryonic stem cells' ability to continue to produce specialized cells generation after generation. So they're not as, typically not as potent or useful as the embryonic stem cells are. So there are some of these adult stem cells, they just can't differentiate as much and for as long as the embryonic stem cells can. As I mentioned, there's essentially 2 different types of stem cell treatment as you're looking at stem cell treatment. There's the stem cell treatment through embryonic stem cells, so from that embryo of a very newly developing human embryo, and then there's production of stem cells through something called induced pluripotent stem cells. Both methods will use reprogramming of cells to yield desired cell types. So this is trying to get stem cell treatment from adult stem cells, and this would be stem cell treatment through embryonic stem cells. In 2007, two research teams developed a type of human stem cell not derived from an embryo. This was, as we mentioned, that second type, the induced pluripotent stem cell, the iPS cell. It was first produced by means of splicing 4 developmental genes into the genomes of an ordinary adult skin cell. iPS cells appear to have all the developmental power of embryonic stem cells. They hold promise of reducing problems of tissue rejection in medical transplantation procedures, and they are widely being viewed as a means of studying human disease. So as time goes on, I think a lot in part to the controversy of embryonic stem cell treatments. They are trying to figure out how to get stem cells essentially from adult tissue and not from embryos. And so this is one possible way of doing this is through this induced pluripotent stem cells and getting stem cells out of adult tissue rather than embryonic cell tissue. Next up, we have section 15.5 on forensic biotechnology. Identities of criminals, biological fathers, and disaster victims are often established today through the use of forensic DNA typing. So there's a lot of different crimes, figuring out family relatives that can be done through looking at someone's DNA. Forensic DNA typing is the use of DNA to establish identities in connection with legal matters such as crime. And the polymerase chain reaction is a technique for quickly producing many copies of a segment of DNA. So PCR, polymerase chain reaction, works through taking a double-stranded section of DNA. So you select the DNA region of interest. The DNA is heated, causing the two strands to separate. As the mixture cools, so short DNA sequences called primers are form base pairs with the complementary DNA sequences in their respective strands. The DNA polymerase goes down the line, synthesizing the complementary DNA strand, so it makes a new strand onto these pieces, and the end result is a doubling of the original DNA. The process is repeated many, many times, doubling the amount of DNA each time. So PCR is essentially a way of taking one copy of DNA and quickly making multiple copies of a certain segment of DNA. PCR is useful in situations such as crime investigations when a large amount of DNA is needed for analysis, yet the starting quantity of DNA is very small. So you can't always do the full criminal analysis and the testing that needs to be done on just a single piece of small DNA. So in a crime scene, for example, you might have a very minute amount of DNA. PCR is a method of quickly making a large quantity of DNA. When you hear about DNA analysis, the way that this is typically done to analyze and compare crime scene DNA to suspect's DNA is through a technique called gel electrophoresis. So the first step that needs to be done when you have crime scene DNA and suspect DNA is you copy the DNA using PCR. So you may have a very small sample of DNA. You make copies of that DNA, so you have a lot of copies to work with for this process. So you need a certain amount of DNA for this process to be able to work. You then go along and you cut the DNA with an enzyme that cuts the DNA when it reaches a certain set of codons. So it sees every time it sees a certain pattern, it makes a cut. So for example, let's say it reaches CGA-ATT, that enzyme will make a cut, and it will make that cut every time it sees that pattern, and it will make that cut in the crime scene DNA and in all the suspect's DNA. Now the difference is that the DNA from different people, obviously you have a different code than DNA code than everybody else. And so your DNA will always get cut at the same place. So you're always going to have this pattern at the same place. And so your DNA will always get cut in a certain pattern. Someone else's DNA will also get cut at these places, the same series of codons, but the way it gets cut will be different because when they reach this pattern, it will be at different places than when it reaches that pattern in your DNA. So the next step is to pour gel into sort of a well and let that gel harden. And they put sort of a comb into that. And this is going to be the basis for your week 8-9 lab. And so you make these holes in the end of the gel, and then you put all of your DNA that's been copied and cut into these wells. So you might put your crime scene DNA into one well, and then all of your different suspect DNAs, if you had several suspects, into the other wells. Then they turn on an electric current. So they hook up a negative current here and a positive current here, and the DNA is very slightly negative. And so when they turn on that current, the DNA that's sitting here in this well, so they've loaded it into the well, and they turn on the current, and this DNA is going to be attracted to the positive end. If it's slightly negative, it's going to be attracted to a positive, and the DNA will start moving through this gel. They let the DNA pieces move very slowly, but small pieces will move a little quicker and large pieces will move more slowly. So if you think of trying to move a large object versus a slow object, the large objects just are heavier and they move more slowly. The small pieces of DNA move more quickly. And so each person's DNA fragments, since they were cut here, but everyone's DNA is a different pattern and got cut in different places when it reached this, different people's DNA will move at different speeds. And so what they will do is they will compare the pattern of the crime scene where the DNA pieces move to all of the different suspects. And ideally, if they had the right suspect, they could see that, for example, if this was the crime scene DNA here, that suspect 3 here on the right, say this is suspect 1, 2, 3, suspect 3 here matches the DNA pattern. And what you would be looking for is an exact match, so not something similar, but an exact match in where that DNA moved. And if you had an exact match, it's very, very highly likely that that DNA came from the same person. So it's really, really very, very unlikely that the DNA from 2 different people would match up when doing gel electrophoresis if they were not the same person. And so that's the process that a forensics lab will use to help police and determine, analyze crime scene DNA versus suspect DNA. Your book talks about the case of Earl Washington, who was convicted and sent to prison for murder. And later they analyzed DNA from semen on the victim and from this man, Earl Washington, and from another man who they later thought might have done the crime. And they found that when they looked at each of these places in that DNA well and where those locations occurred, that the measurements for Earl Washington did not match up, as you can see, to the semen on the victim. And they did match up for Kenneth Tinsley. So after being in prison for 17 years, Earl Washington was released using DNA evidence to prove that he did not commit the crime. We've talked a little bit on the controversies in biotechnology. So far, let's take a look at a few more of the controversies that are issues in biotechnology right now. So first off, biotech progress also comes slowly because so many of the processes it's developing are not just new, but they're also controversial. So sometimes things are developed, but they are slow to get put into use because their health and safety and ethics are controversial. So a notable biotech controversy concerns genetically modified crops. So we can modify foods in many different ways, but there's the issue of whether it's healthy to do so. So in general, the FDA in our country does a certain amount of testing and accepts foods as safe if and allows them for production if no negative impacts have been observed so far. Other countries such as Canada and countries in Europe take a much more conservative approach and will only accept foods as safe once they are proven after a long period of time safe. So after lots of research and lots of time, if they are proven safe, they are accepted. The US tends to accept them if they haven't seen anything bad so far. So it's sort of a difference in acceptance. But in general, the US accepts many more things for allowing them to be used than other countries, the UK, Europe, Canada. They do not accept as many genetically modified foods as they. So opponents of genetically modified crops are concerned about their effect on human health and the environment. So you can make foods, for example, with built-in genes that create pesticides to kill certain insects. So rather than spraying, the chemical is built in. One example of this is a gene from a bacteria called Bacillus thuringiensis, and this is a bacteria that produces a toxin that kills certain insects. And this toxin is built into the plant, so you don't wash it off, it's built in, and it produces these toxins that kill the insects in the plant that gene is inserted into. So for example, US corn is produced with that gene built into it, and this gene can also be placed in cotton, and we'll see in the next slide. And places like the UK do not allow the growing of any GM foods. So they would not allow corn with this gene to be placed into it. They're concerned with the safety for human health and also for the environment. So that's just a look at differences in different countries on what foods are accepted. And certainly that's a controversial issue of whether we should be having modifications such as this to our food. Now here is a picture. The reason that the controversy comes up is that this built-in gene is very effective. So you can see cotton plants on the left grown with this gene. The cotton is growing very well. This cotton over here that's gotten attacked by insects, same field, but this cotton on this side does not have that gene, this Bt gene in it, the gene that's from the bacteria that produces toxins, and this cotton was attacked by insects. And so inserting this gene is and modifying the plants in this way is very effective. The question is just whether it's safe for our environment and humans to do this. So looking at something of whether to genetically modified food is an issue more of environmental safety and human health. Some biotech controversies like we've been talking about are much more ethical in nature. So among these controversies are things such as using embryonic stem cells and therapeutic cloning. So whether you should allow cloning or use of stem cells as treatments. And these are more ethical. So I'm sure there's health impacts as well. But the issue is much, the issue people have with it is more of an ethical issue than a health issue generally. Because these are such controversial topics, there are a lot of people on both ends of the spectrum who have very strong opinions. And I think the most important thing as you go about making decisions on what things you vote for, what foods you eat, what medical treatments you would seek, is that you are informed and you make informed decisions. So keep in mind that there are going to be people with very strong opinions. Look for, as you're reading, look for articles and ideas that are not just people's opinions, but that are backed up with scientific evidence or good ethical discussions, depending on what the issue is. And you'll make the decisions on these issues that you feel are right. And I don't think on any of these there's any one right answer for everyone. People will definitely have different opinions, and that's okay. The important thing is to know what you feel is right in these cases and make the best decisions based on what you feel is best. All right, that's the end of our Chapter 15 lecture, and please let me know if you have any questions as you go about the last couple weeks of the class.